Hello, everyone. My name is Wendy Semsky, and I'm a PhD student at the University of Wisconsin-Milwaukee. Today, I'll be talking about individual flowering schedules and floral display size in monkeyflower. Flowering patterns often vary widely within and among populations and can have important consequences for reproductive success. The extent of flowering overlap can influence pollinator attraction, as well as the number and quality of mates. Two important components of flowering phenology are the date of flowering onset and the length of the flowering duration. These figures show flowering intervals for 5 individuals from 4 imaginary populations. The two populations on top differ only in the date of flowering onset, and the two populations on the bottom differ only in the length of the flowering duration. Simultaneous flowering onset results in complete overlap, maximizing mating opportunity since every plant is available for pollination. However, as variation in the dates of flowering onset increases, mating opportunity is more limited. This effect can be mitigated as the duration of flowering increases, leading to increased overlap of flowering intervals. How flowers are deployed throughout the season can also influence pollinator attraction, as well as the extent of among-flower self-fertilization. Plants may differ in patterns of flower deployment, both within and among populations. For example, these figures represent the flowering schedules of 4 plants from 3 imaginary populations. Each row represents an individual, and each circle represents a day on which the plant flowered. The size of the circle corresponds to the size of the daily floral display, so the larger the circle, the more flowers on that day. In population 1, flowering schedules are nearly normally distributed, and plants deploy their largest displays simultaneously. In population 2, however, flowering occurs in pulses, so that peaks do not occur simultaneously. Finally, in population 3, there is very little day-to-day variation in floral display, so flowering is steady over the season. An important thing to note here is that the total flower production does not vary. All 12 of these plants have the same total number of flowers, but they differ markedly in how they are deployed. Although large daily floral displays may enhance the attraction of pollinators and promote pollen export and receipt, they may also experience higher levels of among-flower self-fertilization. This is because pollinators tend to visit multiple flowers sequentially on a single plant. This type of selfing is costly because pollen and ovules that would otherwise be available for outcrossing are instead used for selfing. In populations with substantial inbreeding depression, selection should therefore favor plants that produce small floral displays over many days, rather than plants that produce large displays over few days. One challenge in studying variation in floral display size is that the number of open flowers per day depends on both daily flower deployment as well as floral longevity. In other words, the daily floral display consists of flowers that opened today, as well as flowers that opened previously. It can be difficult to disentangle the relative influence of these factors. Approximately 27% of flowering plant species have flowers that last for just a single day. These species can be very useful in understanding how patterns of flower deployment shape individual flowering schedules, and how attributes of their flowering phenology influence total flower production. Monkeyflower, Mimulus ringens, is especially well suited for answering these questions. It is a hermaphroditic perennial native to wetlands in North America. It produces purple flowers, each of which lasts for only a single morning, so floral displays do not overlap between days. Monkeyflower is primarily bumblebee pollinated. Every flower produces a fruit, and it exhibits a mixed mating system, so populations differ in their mean selfing rate. In 2018, we surveyed 9 natural populations of monkeyflower in Northeast Ohio. We found wide variation among populations in total flower number, which ranged from a mean of 19 flowers per plant to a mean of 85 flowers per plant. There was also striking variation within populations in flower production. For example, in some populations, most plants produced few flowers in total, while in other populations, total flower number varied widely among individuals. And so my talk today will investigate whether this variation in flower production is also present in a common garden, how it is related to other components of flowering phenology, and also how synchronously individuals deploy their flowers across the season. By raising plants in a common garden, we can control for environmental variation and determine whether these differences have an underlying genetic basis. To answer these questions, we raised 20 progeny from seeds collected from the plants measured in our 9 natural populations, taking special care to minimize environmental variation. Seed germination was highly synchronous across populations. When plants began flowering, for every individual, we noted the date of flowering onset as the date on which the first flower opened. Then, every other day throughout the flowering period, we quantified every plant's daily floral display size. And so this means that we tracked the flowering schedule for every individual. We use those data to quantify components of flowering phenology, such as total flower number, flowering duration, and flowering synchrony. In this figure, the x-axis is the date of flowering onset based on the Julian calendar, and the y-axis is total flower number. Each point represents an individual and is colored according to the population to which it belongs. We found that the date of flowering onset is negatively correlated with total flower number. Plants that begin flowering early tend to produce more flowers than plants that flower later, so earlier flowering leads to greater fecundity since every flower produces a fruit. Additionally, we discovered that this pattern is maintained within populations and is significant in most of them, as you can see in the table on the right. This figure shows variation in the date of flowering onset for our 9 populations raised in the common garden. Date of flowering onset varies widely, both within and among populations. For example, in population LNB, the shape is short, indicating that flowering onset occurred over a very brief window. In contrast, in population MCW, the date was much more variable amongst individuals. Similarly, we see in the figure on the right that total flower number also varies widely in the common garden. In some populations, most plants produce relatively few flowers in total, while in others, total flower production is more variable and is evenly distributed among individuals. We also find that flowering duration is positively correlated with total flower number. In this figure, the x-axis is the length of the flowering duration in days, and the y-axis is total flower number. Each point represents an individual and is colored according to the population to which it belongs. Here, plants that flower longer tend to produce more flowers than plants with a shorter flowering duration. This pattern is supported within the populations as well, which I show you in the table on the right. And we also found that the length of the flowering duration is highly correlated with date of flowering onset. And so taken together, these results indicate that plants that initiate flowering early are also able to continue flowering, and this results in greater fecundity. And so now, let's take a closer look at the daily experience of plants and how their patterns of flower deployment influence flowering synchrony. Earlier, I showed these figures, which represent the flowering schedules of 4 plants from 3 imaginary populations. As a reminder, each row is a plant, and each circle is a flowering day, so that the size of each circle corresponds to the size of the daily floral display. Previously, I explained how variation, or lack thereof, in the shape of the individual flowering curves contributes to population level patterns. Now, I want to highlight how daily variation in flower deployment can affect mating patterns among individuals. This idea is best illustrated in population 2, where plants deploy their flowers in pulses so that flowering is more sporadic across the season. As a result, the number and identity of plants in flower differs from day to day, and thus, how synchronously individuals deploy their flowers influences their mating opportunity. We can quantify the extent of flowering synchrony among individuals in each population to determine the extent to which flower deployment varies across the season. This figure shows the flowering schedules for every individual belonging to population MSB. The x-axis is Julian date, so that day 190 is July 9th. Each row represents an individual, so that each circle represents a day on which that plant flowered, and plants are organized along the y-axis by their date of flowering onset. As before, the size of the circle corresponds to the number of flowers in a plant's daily floral display, so the larger the circle, the larger the floral display. In this population, individuals appear to flower sporadically with large day-to-day variation in their display sizes, and the largest floral displays do not occur on the same day. Also, I'd like to point out one day in particular, day 206. What's striking here is that although this day occurs in the middle of the flowering period, only about half of the plants in this population are flowering. This is especially interesting to see in a common garden, since asynchrony is likely to be greater in the wild due to environmental variation. We used Augsburger's synchrony index, which ranges from 0, no synchrony, to 1, complete synchrony, to quantify the extent of flowering synchrony among individuals in each of our 9 populations. And here I show you the results for just 2 of them. Population MSB, the figure on the left, had the lowest synchrony, while population RIS, on the right, had the highest synchrony. This is especially apparent when we compare day 206 between these 2 populations. As we saw, MSB has only about half of the plants in flower, while in RIS, every plant is flowering. In RIS, flowering peaks appear to be larger, and many plants are quite synchronous in the timing of their largest displays. In this common garden study, we found wide variation within and among populations in the components of flowering phenology, as well as in patterns of flower deployment. Plants that flower earlier tend to flower for longer, which results in greater fecundity. By measuring flowering schedules across the season, we can gain insight into population attributes such as flowering synchrony and the timing of the flowering peak. Thank you very much for listening.